All right. So I'm going to introduce our next speaker. It's Dr. Susanne Brander. She's an assistant professor at Oregon State University. She's an ecotoxicologist whose research integrates responses of aquatic organisms to endocrine disrupting compounds and other environmental stressors such as microplastics across the biological hierarchy. So it is a pleasure to have you, Dr. Brander, and I'll turn it over to you so you can talk about risk assessment.
Great. Thank you so much, Shelley, and thank you for having me today. Like was explained, I'm going to be talking about microplastics risk assessment. I'll be going over several processes underway on the West Coast, two that are underway and now near completion in the state of California, and a third that is a collaboration between OSU, Western Washington University, and some of the great work being done at the San Francisco Estuary Institute, all to assess the risk of microplastics in ambient waters. And the work I'll be talking about today is part of several very large collaborations and is the result of the contributions of many. So I just want to acknowledge that before I begin. So first, I'll give a brief introduction on the toxicity of microplastics and why we're concerned about risk. I'll talk briefly about the California Ocean Science Trust report that came out earlier in the year in the spring. We'll talk mostly about the California Microplast Health Effects Working Group that was convened by the Southern Coastal California Water Research Project and the California Water Boards, and then talk a bit specifically about Bayesian modeling approaches that are currently underway between my research group that I co-lead with Dr. Stacy Harper at OSU, which is the Pacific Northwest Consortium in Plastics, and some work we're doing in collaboration with SFEI. So what responses are seen to microplastic exposures? And others in this webinar today have referred to these responses here and there, but we see a myriad of different effects in aquatic organisms. We, as a large research community, are beginning to converge on some commonalities here. Two in particular that I'll focus on today are impacts on growth, likely caused by food dilution or false satiation when an animal ingests a microplastic, as well as oxidative stress, which may occur due to translocation. It's also important to acknowledge that some studies see no effect, and often impacts are only observed at high concentrations. But we are beginning to see these common responses across different taxonomic groups, as well as across biological scales. What do we know about toxicity? To briefly discuss this, we know that size and shape influence toxicity in laboratory studies, and that evidence shows that fibers may be more toxic than other plastic shapes or morphologies, and that the smaller the microplastic is, and even getting down into the nanoplastic size range, so below a micron in size, those smaller plastics appear to be more likely to cause oxidative stress. Upon exposure to microplastics, cells can produce reactive oxygen species, and then these ROS can cause structural damage, and some of this may be due to translocation. And again, like I said in the last slide, food dilution and translocation appear to be major drivers of toxicity in aquatic organisms. But of course, we all know that we can see plenty of responses and indications of toxicity in laboratory exposures, but those responses don't necessarily reflect what's happening in the wild unless that exposure is in the lab is relatively similar to what's happening in the field. And so it's really only when that hazard and exposure overlap that you're able to accurately estimate risk. And that's the aim of several of these efforts that I'll talk about today. So two state-level actions in the state of California were the Ocean Science Trust Science Advisory Panel, and this began right at the start of the pandemic in spring of 2020, and this was a qualitative risk prioritization framework that was put together specifically for marine ecosystems in the state of California. Report came out earlier this year, really relied on the precautionary principle, source reduction, so limiting the inputs of plastics into the aquatic environment was emphasized. This was all convened by the Ocean Protection Council and grouped scientists scientists from the US and Canada to work on this issue. And then the Microplastics Health Effects Workshop, which is a much larger effort, was convened by SCWRP and the water boards, and this involved experts from Europe and North America. It resulted in the creation of the TOMEX database, so data from 167 different peer-reviewed papers, and I'll talk about the species sensitivity analyses that were done from those data. And then the goal of this was threshold derivation for ambient water and for drinking water, although today I'm just going to focus on ambient water in the interest of time. And so in response to State Bill 1263, which Dr. Coffin referred to in his presentation, the group convened by the Ocean Science Trust came up with a risk characterization and ranking process. And again, this is qualitative, and we were a bit limited because the focus was primarily on the state of California and on species of concern in the state of California, for which right now there's really pretty limited data. But in the meantime, we came up with an approach that includes some key factors to be considered, such as particle morphology, polymer types, priority species or endpoints, fate and transport, and sources. And the outcome of this is that one can use this framework to characterize and rank risk based on the amount of microplastic that is estimated to be loading into the environment and the risk of ingestion or internalization by priority species. And then, of course, because we're limited in those data for California species, surrogate species can also be used. So it basically prioritizes source reduction strategies and allows consideration of risks that might warrant state action or potential mitigation. For the Microplastics Health Effects Working Group, which was convened again by SCWRP and the water boards, this involved scientists from North America and Europe, and tasks for the group were to agree on a management framework for microplastic assessment in ambient waters, derive a method to produce thresholds, species sensitivity distributions were used, and I'll talk a bit about this, and all of the studies considered holistic analyses underwent very strict quality assurance and quality criteria. Be familiar with, but have been used for decades. They're a probabilistic method to derive thresholds for toxicity of a variety of different environmental stressors, different groups of contaminants. Often values for algae, crustaceans, mollusks across a number of different taxonomic groups are used, usually relies on laboratory testing. And the goal here is to calculate an estimated hazardous concentration for a large majority of species. Usually the cutoff is at an HC5 or an HC10. So you're protecting either 95 or 90% of species in that SSD. For the Microplastics Health Effects Group, separate sets of thresholds were derived by aligning data based on two primary mechanisms that I talked about a little bit earlier. The first was food dilution, which can result in limited growth and other impacts like limited reproduction, and then tissue translocation, which is thought to drive these oxidative stress responses that are being seen in many different types of organisms exposed to micro and nanoplastics. So I certainly don't expect you to take all of this in. There's a lot going on this slide, but these are the 4 species sensitivity distributions that were derived from this ambient water working group. The ones up top were used for food dilution, and after QA/QC was performed, we ended up with studies that represented 15 species across 6 different taxonomic groups. And then at the bottom, these are the species sensitivity distributions used for tissue translocation. And here, after QA/QC, we ended up with 16 species and across 6 taxonomic groups. So enough of a representation across taxonomic groups and studies to derive accurate SSDs. And another important thing to mention is that, of course, most of the laboratory studies that were in the database being used for this effort are monodisperse or people were exposing animals to one type and one size of microplastic or nanoplastic. And so these monodispersed concentrations were rescaled to a default polydispersed size range of 1 to 5,000 microns. And then if the particles were larger than the organism in question's mouth opening or were too large to translocate, those particles were excluded. So the ultimate goal here was to come up with a decision framework that could be used by the state to evaluate microplastics as a stressor. The framework you're seeing here is similar to past frameworks that have been used for other chemicals and other stressors. And the development of this management framework really requires generation of analysis that informs several thresholds, and each of those thresholds is linked to some sort of management action. And the lower thresholds are meant to be protective of effects on aquatic organisms, and so exceedance would require something more minor like investigative monitoring. And then as you get to those higher levels of concern, that's where it would lead to more costly management actions such as mitigation. So earlier decisions are inexpensive and reversible, and later steps have a larger cost. And all of this work is currently in prep in a manuscript being led by Dr. Alvina Mahinto from SCORP. These are the species sensitivity distribution parameters and data filters used to derive thresholds for the tiered framework shown in that previous slide. So again, HC is hazardous concentration. The 5 represents 5% of species, and so the HC5 would be protective of the other 95%. You need to make that cutoff somewhere, especially since when you have a species sensitivity distribution, you have a lot of variability in that lower side, those lower endpoints. And you can see as one moves up the threshold, the criteria need to exceed that threshold are increasing. So we go from HC5 to HC10. We go from including molecular to population level endpoints to just including organism and population level endpoints as you get higher up those thresholds. For food dilution, we are estimating thresholds for food dilution as well as for translocation and oxidative stress caused by translocation. For food dilution, there's a big difference in threshold calculations depending upon whether one includes all of the endpoints, only fitness-related endpoints, so things that are going to affect an organism's ability to survive and reproduce, or if you limit it just to mortality, those concentrations do get much higher. And just to put this into perspective, 0.2 particles per liter for that threshold 1 seems like a very low threshold, but if you're thinking about this from the perspective of a smaller organism like a daphnid or a larval fish used for aquatic toxicity testing, that's roughly 200 particles per milliliter, which isn't as low as it sounds when you're putting it in particles per liter. Similarly for translocation, and remember this is for particles that are 83 microns in size or smaller, and that's the estimate of how small a particle needs to be in order to translocate within an aquatic organism. Threshold values also increase here depending upon whether all endpoints are considered versus just fitness related or only for mortality. And really in microplastics studies and lab studies, mortality is fairly rare. Usually you're seeing sublethal responses. At present, reported concentrations for both the translocation and the food dilution SSDs are lower than these derived thresholds, but plastic production is predicted to increase exponentially by 2050, and thus if we continue business as usual, as predicted by several recent publications, this will likely lead to higher environmental microplastic concentrations. So it's good to be a bit precautionary here and to think out to what future exposures might look like for aquatic organisms. So the vast majority of studies in the TOMEX database, and again there are 167 studies total, and this was Great work done by Dr. Leah Thornton-Hampton at SQUIRP and Dr. Scott Coffin, pulling together all of these data that are being used both for generation of the ambient thresholds as well as generation for screening levels for drinking water, which I'm not going to discuss today. But the vast majority of the studies in this TOMEX database are done on spheres and fragments. Spheres in particular are not necessarily representative of what is detected in biota. As someone who goes out and samples fish and other organisms, I can definitely report this from experience, and you heard this from Dr. Lin at SFEI. And as you can see here, there are only 5 studies currently in the TOMEX database on fibers, which is what we really mostly find. And the number of studies in fibers is expected to increase as more data are added. And I show this to say that it's important to remember that the main goal of this work was to build a framework for the assessment of risk, and those thresholds will likely become more accurate and reliable as additional data on more realistic plastic types like fibers are included. And so just to hit this point home, thresholds may change as additional data are added because we know that fibers, both synthetic and natural but of anthropogenic origin, are ubiquitous in water, sediment, and biota globally. And more studies on fibers have already come out since the database was completed and are going to be added as they are published, and there are many more in progress. So moving past the microplastics health effects group and getting into some work that is specific to the Pacific Northwest Consortium on Plastics, which I co-lead with Dr. Stacy Harper at Oregon State University, and we also have Dr. Wayne Landis from Western Washington University, who is a PI on this grant, and all of this work is funded by the National Science Foundation Growing Convergence Research Initiative. And Dr. Wayne Landis's group, and specifically his current graduate student, Emma Sharp, focus on deriving risk assessments using a Bayesian network approach. So what is a Bayesian network? Well, I'm showing kind of a simplified version of that here. It's graphically depicted here is a web of nodes that link cause and effect relationships using conditional probability. And it's a way to describe interactions and to generate the probability outcome, or more often outcomes, multiple outcomes, so it allows for a lot of complexity. Another benefit of this is it's habitat specific, and so you're seeing the map here of San Francisco Bay microplastic risk regions, and the Bayesian modeling approach allows you to calculate risk for specific risk regions that are including characteristics of habitat, potentially land use that's specific to that area. And so it's a different type of approach from the species sensitivity distributions that were derived, in that the SSDs are a bit more generalized, where this can be specifically sort of targeted to a particular region. And in this case, because of SFEI's really groundbreaking study, there is a lot of great data to be used in order to inform a Bayesian modeling approach. And so Dr. Landis's lab and the rest of our group have been collaborating with SFEI, basically combining data from the lab with great data from the field. And just as a representation for one of these nodes, you're seeing one of the nodes in this network could be a laboratory study, generating data on growth or swimming behavior or mortality, for example, in a representative species. So, try not to spend too much longer on this since we're a little bit short on time. But what I'm showing here, and this is a lot to digest, is a Bayesian network for tire wear. And this is a preliminary risk assessment framework. And basically what you're seeing here is that everything starting from sources all the way out to impacts is included in this modeling framework. And under impacts are species of concern that can be impacted by a combination of factors. And again, this is work being led by Emma Sharp, pictured at the bottom there. She's a graduate student at Western Washington University. So Low, medium, and high risks are based on the percentage of endpoint species of concern on the far right that are impacted by a combination of factors, in this case including tire wear particles, but also including other considerations like land use, transportation, ecological factors, and even whether it's the rainy versus dry season, for example. How does this all work? Well, each node can have up to 4 different states in this particular example, and then the percent of values in each discrete bin is predicted by the model. And so this is a Bayesian net example that's kind of hot off the presses, and this is work that's underway right now. And you can see we have the San Francisco Bay microplastic risk regions over on the left here. In the middle is data that is mainly being generated by the San Francisco Estuary Institute. So this is tire wear particle field data that has been collected from stormwater, sediment, effluent, and in surface water. And then over here on the right, we have lab data that is mainly being generated at Oregon State University by my lab and the Harper lab on a variety of different aquatic organisms. We also have some work being done out at the Hatfield Marine Science Center by the Langdon Lab on larval oysters, and so that work will eventually be included as well. And so as we select different inputs in one node, the distributions change in the rest of the model, and there are a lot of benefits to this. For example, you can have the model be reliant only on one of those risk regions. And so here, you're saying that 100% of this model is reliant on the San Francisco Bay region. And then you can also work backwards by changing the nodes at the end. For example, you could calculate what input would cause, say, a 20% increase in daphnid mortality. And then the ultimate goal is to use some of these surrogate species and data that we're getting from the University of California, Davis on internalization of microplastics in salmonid species to predict out to species of concern like Chinook salmon, which we know are affected by tire wear leachate. A study from Washington State was published earlier in the year on that, on coho and Chinook. So to sum this up and to make sure I give our last speaker enough time, the Ocean Science Trust Report has been finalized, is now public, is making its way to legislators in California at this time. Publications from the Microplastic Health Effects Working Group will be out in the coming year. And research recommendations from these efforts will certainly lead to the collection of data that better supports further risk assessment for microplastics. And from our group, the Bayesian model for tire wear particles is estimated to be completed soon. And then we will add in assessment of other particle types. There's testing with fibers, for example, and other plastic fragment types underway in labs at OSU at the moment. And I'll sum it up there, and thank you very much for your time. If you'd like more information on our work, you can check out our website, and we're also on social media. And just thank you very much to all of the students and to my postdoc, Dr. Samreen Siddiqui, who we couldn't do any of this work without.
Thank you, Suzanne.